If you look at this slide here, this simply shows you how the gametes are formed during meiosis, and there's a focus on the case where we're dealing with more than two characters at once. So, for example, we might be dealing with the G character, which could be maybe color of something, or even just size of something, and the Y character, same thing, color or size. The point is we're trying to show this is dihybrid. Basically, what's happening here is you can see that the G's separate, right? So like when you have the gamete over here, the big G's are in their own gametes, and the little G's are in their own gametes. And the same thing with the Y's, they separate. The little Y's are in their own gametes, and the big Y's are in their own gametes. And the reason for that is because the same letter, just different casing, means that they're part of homologous chromosomes. They're alleles. They're different versions of the same gene. So this might be yellow. This might be green, the gene that controls for color, let's just say. And so that's the law of segregation, which basically means that the alleles have to separate during gamete formation because the homologous chromosomes separate. Like this small one goes this way, this small one goes that way. So the alleles go with them. So that explains why you have only one type of Y in a gamete, not both types, and only one type of G in a gamete, in this particular one. Right? You're never going to see a big G, little g in a gamete. So that's the law of segregation. The law of independent assortment explains why you can have so many different types of gametes. So if you look at big G, big Y, little y, you can have big G, big Y, big G, little g, big Y, and little g, little y. If you look at this picture, it doesn't quite show that. If you look at this picture, we just have big G, little y going together, so you have big G, little y. That's it. And here you have little g, big Y going together, so you have little g, little y. Or little g, big Y, sorry. Okay, well, where are the other ones? We have big G, little y. We have little g, big Y. So where do these come from? Well, that comes from the idea of the law of independent assortment. If you look over here, this is metaphase 1. The big G and little g paired up. The little y and big Y paired up because they're homologous. And they pair up along this metaphase 1 plate over here. And so what's important to understand is that big G did not necessarily have to be on the same side as little y. Instead, this could have happened. Instead, big G and big Y would have been on the same side of the metaphase 1 plate, and little g and little y are on the same side as of the metaphase 1 plate. And so that gives you your little g, little y gametes and big G, big Y gametes, which we said you could make over here. So this slide simply shows how the gametes form in the process of meiosis. Obviously, when you have to do a quick Punnett square, you won't need to draw this out every time, but it's good to know how to draw it out to explain it. When you're doing a quick Punnett square, you just do big G, big Y, big G, little y. Little g, big Y, little g, little y. But to explain why the G's don't end up together, you would say the law of segregation, because homologous chromosomes split up and the G's are found on homologous buddies, pairs. And same thing with the Y's. But to say, why you can get different combinations, big G with big Y or big G with little y, you would explain the law of independent assortment. And that's because of the way they arrange themselves around the metaphase 1 plate. The G's have to be paired up and the Y's have to be paired up, but there's no expectation that big G needs to be on the same side as little y or big Y. They don't care what the other pairs are doing. So you could have this arrangement or this arrangement, and depending on what you get different gametes. So big G, little y, little g, big Y, big G, big Y, little g, little y.